Hello everyone, my name is Binay. Welcome to my channel LifeLore. Today we are going to discuss about hematopoiesis. So what is hematopoiesis? Hematopoiesis is the production of different blood cells from hematopoietic stem cell. So this whole process is called hematopoiesis. So where does it happen? It happens in bone marrow. So this is the bone marrow. So it starts with hematopoietic stem cell. So this is hematopoietic stem cell. So usually what happens in bone marrow? Hematopoietic stem cell. What is the stem cell? Stem cell is a kind of cell which can give rise to different types of cells. So this is multipotent. So usually in bone marrow, stem cells also, there always a renewal process. So this stem cell always renew to a new stem cell. This happens normally in the bone marrow. And then stem cell becomes progenitor cells. This is also multipotent. This process is one-way process. Once it becomes progenitor cells, it cannot go back to hematopoietic stem cell. From hematopoietic stem cells comes the progenitor cell, hematopoietic progenitor cell. From here, it goes either common myeloid progenitor cells or common lymphoid progenitor cells. So from common myeloid progenitor cells, it gives rise to two kinds of cells. One is megakaryocyte erythroid progenitor cell, and another one is granulocyte macrophage progenitor cell. So from megakaryocyte erythroid progenitor cells, these erythroblasts differentiated, and from erythroblasts these erythrocytes are coming. So this part we will talk about in our next video. So please subscribe to the channel. And also from megakaryocyte erythrocyte progenitors, megakaryocytes are coming. And from these megakaryocytes, platelets are coming. And granulocyte monocyte progenitor cells, they become granulocytes like mast cell, basophils, eosinophils, and neutrophils. And also they become monocyte dendritic precursor cell. From this precursor cell, dendritic cells are differentiated. And from these precursor cells, monocyte cells are developed. These monocytes then become macrophages. And this part is myeloid. This total part is from common myeloid progenitor cells, and next common lymphoid progenitor cells. And from these common lymphoid progenitor cells, B cells develop, and from this T cell, NK cell, precursor cells develop, and from this T cell and NK cells are coming. And these T cells usually are formed in the thymus. The differentiation and maturation of these T cells happen in thymus. So this is the common and the brief process of hematopoiesis. This happens, hematopoiesis, the main part of the hematopoiesis happens in bone marrow. After the differentiation, they transported and they come to the blood. So as I said earlier, this is hematopoietic stem cell. From hematopoietic stem cell, different kinds of cells are arising. So this is after becoming differentiated, they cannot go back to the hematopoietic stem cells. This is the one-way process, but hematopoietic stem cell can give rise to another hematopoietic stem cell. So because this is the natural process, we can do this process in vitro. That means in your lab. So for that, we need to isolate hematopoietic stem cells. So hematopoietic stem cells either are isolated in human case. Hematopoietic stem cells are isolated from cord blood cell. So after the delivery of the baby, the cord blood is collected. So from cord blood, this is from umbilical cord. After cutting the umbilical cord, the blood is collected, and there is a heavy chance that there will be sufficient hematopoietic stem cells in the cord blood. So from cord blood, we isolate hematopoietic stem cells. So I'll talk how we isolate this. This is in human case, and we need to get the consent from the donor also in this case. And in mouse, how do we collect hematopoietic cells in mouse? Suppose this is one mouse. So suppose this is one mouse, and we isolate the femur, the bone marrow, femur from the mouse. After isolation, we flush this bone marrow, and this is the femur. We use syringe to isolate this bone marrow, to flush this bone marrow, and then after flushing, we separate this bone marrow, mononuclear bone marrow, using histopack or gradient centrifugation. So usually when we centrifuge these cells, we found mononuclear cells in the middle. You'll see those mononuclear cells will look whitish, and you can isolate these mononuclear cells. So after isolating these mononuclear cells, now is the time we find out our hematopoietic stem cells. So in both the cases, in human and also in mouse case, we use flow cytometry to identify these hematopoietic stem cells. So usually we use CD34 positive cells, we use CD34 antibody along with lineage. We need to isolate lineage deficient cells. We do not need lineage. These are the lineage cells. If we need to isolate these cells, we need to isolate lineage deficient cells. And also similarly in mouse case we use CKD positive and lineage negative. So we use these antibodies to isolate these hematopoietic stem cells. So how we do flow cytometry, we'll discuss in our next in further videos. After isolation, we put these cells in semi-solid medium like methyl cellulose medium, usually 1%. In methyl cellulose medium, we put these cells and they cannot move. So, because they cannot move, they can form the colony, separate colonies in the plate. So in the plate, we put methyl cellulose medium, and in that we put these hematopoietic stem cells. So after putting these hematopoietic stem cells, they will form, and we'll supply the main ingredient, main cytokine for the maintenance of hematopoietic stem cells are stem cell factor or Steele factor. Second, IL-3, and the third, IL-6. These three are very important for maintenance of stem cells. And apart from this, if you need to make some particular colonies like granulocyte macrophage progenitor cells or granulocyte macrophage colonies or erythrocyte colonies, we need to supplement other cytokines. Like if we need to produce the colonies which have granulocyte macrophage cells, so we need to supplement with colony stimulating factors. So like in erythroid, we need to provide erythropoietin. Like in GMP, we need to provide granulocyte macrophage colony stimulating factor. So these are available commercially. We can buy these stimulating factors and provide in cell culture. So that these cells will give rise to different colonies, and we can study those colonies. So this is the normal process we perform in our lab to isolate these kind of cells. So that is all about hematopoiesis. Thank you for watching my video. Bye bye.